Hello, my name is William Tahaney, and I'm a postdoctoral fellow at MD Anderson Cancer Center. I would like to thank the selection committee for giving me the privilege of talking to you about my research on how the inhibition of GPX4 induces preferential death of p53 mutant triple-negative breast cancer cells. So breast cancer can most broadly be broken down into three main categories. About 70% of women will be diagnosed with what is termed estrogen receptor positive breast cancer, and a further 20 or so percent will be diagnosed with HER2 positive breast cancer. And there is some overlap between these two. The remaining 20 or so percent of women will be diagnosed with triple negative breast cancer, which exhibits a very poor overall survival. But what I want to talk to you about today is the high rates of p53 mutation that are found in triple negative breast cancer, as shown by these two independent data sets. So p53, which is informally known as the guardian of the genome for its roles in the protection of cell cycle, DNA repair, and apoptosis, is a master transcription factor, as you can see. P53 itself begins with a transcriptional activation domain, followed by a proline-rich domain, and then has a DNA binding domain, which is the portion of P53 that is responsible for physically interacting with DNA and inducing these gene transcription changes. You then have a tetramerization domain, followed by a C-terminal domain. So one of the problems with targeting these mutant P53s is the wide occurrence of different mutations. There are some hotspot mutations in p53, such as R175, R248, R273, but as you can see, even these don't crack 10% of the total mutational burden within breast cancer. Moreover, previous p53 specific drugs are only effective for limited mutations and not effective for most mutations. For instance, there's two advanced drugs in clinical trials right now, ARP246 and CODY2. However, these have preferential action for the R175 or G245 mutations. So therefore, there is an urgent need to develop mutant p53 specific drugs with broader efficacy for more p53 mutations. So rather than focusing on finding drugs that could physically interact with mutant p53 or drugs that work on only individual mutations of p53, we wanted to know if there were pathways that, when inhibited, will induce the death of p53 mutant breast cancer cells without inducing the death of p53 wild type cells. The first aim was to identify critical pathways necessary for the survival of p53 mutants and not p53 wild type breast cancer. The second aim to identify the mechanism by which identified pathways can regulate survival and death signals in the context of p53 status. And finally, to determine if this pathway sensitivity is altered by p53 mutational status. And so we chose to identify these critical pathways using high-throughput drug screening. So we performed an in vitro screen where we asked the question, can a drug induce the death of p53 mutant breast cancer? We did this in 8 different p53 mutant cell lines, and if the answer was yes, we then took the drug forward and asked the counter question, which is, does the drug induce the death of p53 wild-type breast cancer? If the answer was no, we then took that drug forward as a candidate. We also performed this in silico by mining the Broad's drug repurposing hub. And we asked the same question, does the drug induce the death of p53 mutant breast cancer? We did this in 19 p53 mutant cell lines. If the answer was yes, we took it forward. Does it induce death in p53 wild-type breast cancer? If it was no, we moved it forward. We then took the intersection of these two, and we found a set of common drugs. As you can see, when we took the in vitro screen and the in silico screen and we integrated them, we had 6 common drugs that were found between these two screens. And these drugs are listed here. We found the peroxidase inhibitor, ML162. We also found 2 cell cycle inhibitors, AZD7762 and MK1775. We found 2 cell division inhibitors, the common chemotherapy docetaxel, in addition to SP743921. And finally, we also identified the proteasome inhibitor, ioximab. And due to its understudied role in breast cancer, we chose to move forward with ML162 and its target GPX4. The first question we wanted to ask was, what was the effect of GPX4 inhibition on breast cancer cell growth? So we plated cells, we treated them with ML162, and we determined the effect on cell number. For MCF7 and ZR75, these top 2 cell lines, these are both ER-positive p53 wild-type cell lines. And as you can see, ML162 did not have any appreciable effect on these. We then looked at AU565, which is a HER2-positive p53 mutant cell line, and we saw that ML162 decreased the growth of these cells. We saw the same thing in Cal51, which is a triple-negative p53 wild-type cell line. Again, we saw that there was a growth suppressive effect. However, when we looked at the effect of ML162 in p53 mutant triple-negative breast cancers, we saw that we did not see a growth suppressive phenotype. We saw a death inductive phenotype. And so I have this summarized here in this table. And what we can see is that ML162 induces the death of p53 mutant triple negative breast cancers. So we next want to know if this was due to an off-target effect of the drug. And so to do that, we created a GPX4 knockout cell line in the triple negative background. We used dox-inducible Cas9 cells, which we then transfected with SGGPX4 or SG control, selected clones, and induced knockout. And as you can see, when we treat the cells with dox, we were able to knock out GPX4, which we do not see in the control cell lines. We then looked at the effect of growth, and we saw that when we take the SGGPX4 cells and treat them with doxycycline, we are able to induce death, and all of the others grow similarly to control. What we can see from this is that the knockout of GPX4 induces death of triple negative breast cancer cells. So the question then became, what was the mechanism of death that occurred under GPX4 inhibition? So to do this, we plated cells and then we treated cells with inhibitors of cell death. So we treated with an inhibitor of apoptosis, necrosis, and ferroptosis. We then treated the cells with ML162 and we counted cells. As you can see here, only FS1, a ferroptosis inhibitor, was able to rescue the effect of ML162. And we saw the same thing in our GPX4 knockout cells, where when you have a knockout of GPX4, you see the cells die. However, when you treat the cells with ferrostatin 1, you can see that that rescues the effect. So here's a general ferroptosis pathway. In this instance, lipids become hydroperoxidated, which are those LOOHs, which are then reduced down to lipid alcohols by GPX4. However, in the presence of metabolism, these end up becoming lipid radicals, which induces ferroptosis. And so what we can see from all of this is that FS1, ferrostatin 1, rescues the effect of ML162 and loss of GPX4. So we wanted to see if we could actually visualize this ferroptotic phenotype. And so to do this, we used a fluorescent molecule known as C11-BODIPI, which fluoresces orange, where there's low levels of lipid peroxidation. However, after ferroptosis, there's high levels of lipid peroxidation, and this probe then fluoresces green. So in DMSO, you can see that we have low levels of this oxidized probe. When we treat with ML162, you can see there is a robust induction of these oxidized lipid subspecies, which is then undone by ferrostatin 1 co-treatment. So we have this quantified, and as you can see, it's a significant induction of oxidized C11 fluorescence, followed by a significant reduction when co-treated with ferrostatin 1. So what we can see from this is that GPX4 inhibition induces death through ferroptosis. So we then wanted to know what the effect of different p53 mutations could be within ferroptosis. And so we treated cells with 4 different ferroptosis inducers. And what we found were that p53 missense mutant triple negative breast cancer cells were the most sensitive to ferroptosis. Surprisingly to us, we found that the p53 null cell line, MDA-MB436, which is triple negative, actually behaved like a wild type cell line. So we wanted to know what would be the effect of taking a p53 null cell line and re-expressing a p53 missense mutant in that. So we did that with this 436 cell line. As you can see, when we treat with dox, we were able to induce the expression of either R248Q or R273H p53 mutations. And when we did that, we saw that the expression of a mutant p53 gene was able to resensitize these cells to ferroptosis. Of course, one of the questions that we had was, what is the effect of ML162 in vivo? So we used a xenograft model for that. We took nude mice and we injected them with MDA231 cells, and we randomized and treated them intratumorally with either vehicle or ML162. These are the individual mouse growth curves for the individual xenografts. And in order to compare these, we took the individual slopes of each individual tumor and we compared them. And we found that ML162 treatment was able to significantly decrease in vivo triple negative breast cancer xenograft growth. And finally, we wanted to know what was the biomarker response to the ML162 treatment. So we took ML162 or vehicle-treated tumors, and we examined the expression of Ki67, the proliferation marker, cleaved caspase-3, the apoptosis marker, and 4H&E, which is a ferroptosis marker. And we found that there was no significant difference in Ki67 or cleaved caspase-3, but we did find that ML162 treatment significantly increased the ferroptosis marker 4H&E, indicating that ML162 is indeed inducing ferroptosis in vivo. So in conclusion, the in vitro and in silico screens identified 6 drugs that induce death in p53 mutant breast cancer cells and not p53 wild type cells. That inhibition of GPX4 with small molecule inhibitors or with genetic knockout induces ferroptosis in p53 mutant triple negative breast cancer cell lines. We also demonstrated that the expression of a mutant p53 gene sensitizes p53 null triple negative breast cancer cells to ferroptosis, and that treatment with ML162 was able to decrease triple negative breast cancer xenograft growth and induced ferroptosis in vivo. And so what we've shown is that ferroptosis inducers represent promising targets for treatment of p53 mutant triple negative breast cancers and possibly other p53 mutant cancers. With that, I would like to acknowledge my lab. I'd like to acknowledge my mentor, Dr. Powell Brown, and then all of our collaborators at MD Anderson, at Baylor, and then also at Texas A&M, into their wonderful screening core and all the help that they were. So with that, I thank you for your attention and would be happy to take any questions.